Welcome to Satori, a system for ontology-guided visual exploration of biomedical data repositories. In this video, we demonstrate an implementation of Satori in the Refinery platform, a web-based visual analytics platform for managing, analyzing, and visualizing biomedical datasets. For this demo, we have imported the Stem Cell Commons, a collection of 199 datasets from studies related to the genomics and epigenomics of stem cells. A dataset contains one or more data files associated with a set of attributes like the file name, creation date, technology, organism, or disease state. Some attribute values are annotated with ontology terms, providing context by specifying relationships. For example, when a file is associated with the ontology term neuroblast, we can infer that it is also associated with the term neural stem cell, which is a parent of neuroblast. Stem cell, in turn, is a parent of neural stem cell. Satori helps us to explore the repository with that information. Here we see a landing page. On the left side of the interface, we see the dataset view consisting of the search input at the top, some filters and sorting options below, and the dataset list. Each dataset is represented by a small snippet called surrogate, containing the title, sharing information, and a keyword in context phrase, showing matched keywords when a search is executed. A dataset summary can be opened at any time by clicking on the double arrow button. It contains a description, the technology and sample sources, analyses, references, and the experimental protocols. On the right is the exploration view, consisting of two main visualizations, the precision recall plot in the top half and the ratio plot in the bottom half. The ratio plot uses the tree map technique to visualize the abundance of annotation terms across the whole repository. The size of an annotation term is defined as the number of datasets that have been annotated with that term at least once. The color of a rectangle indicates the depth of the subtree below it. Nodes in light gray without any border symbolize terminal nodes. The precision recall plot also visualizes annotation terms but uses a node link diagram to display terms as nodes. Links between nodes characterize parent-child relationships, where the parent represents a superclass of the child. For example, chemical component is a parent of biotin. Each node is superimposed with a bar and a line indicator. Here, the bar represents the precision of a given term, which is the number of currently retrieved datasets annotated with that term divided by the total number of retrieved datasets. The superimposed vertical line indicates the recall of a term, which is the number of retrieved datasets annotated with that term divided by the overall number of datasets annotated with that term across the whole repository. Here, recall is 1 for all terms because we're looking at the overall repository. All three views are interlinked. Whenever the user hovers over a term in the ratio or precision recall plot, the identical term in the other plot will be highlighted. The datasets annotated with that term are also highlighted in the dataset list. In the same way, annotation terms in both plots are highlighted when the user hovers over a dataset. Let's assume we want to find all datasets related to leukocyte. Let's search for it. Nothing found. We might be disappointed, but keep exploring by native cell type. We see that the most prominent cell types are somatic, animal, and precursor cell. We are sure that a leukocyte is definitely not a precursor cell, so we exclude datasets related to it. We inspect the next column and find leukocyte with a precision higher than 0, so we query for leukocyte. We find 5 datasets. Since we want all leukocyte datasets, we have a quick look at the recall. We notice that we only found less than half of all leukocyte-related datasets in the repository. Maybe some of the datasets related to leukocytes do contain other files related to precursor cells, which we excluded earlier. Since we queried for leukocyte anyway, we can't find unrelated datasets. Now we see that the recall went up to 1, meaning that we found all datasets related to leukocytes. Next, we remove the routing to a native cell to explore which species of data the datasets are associated with. It turns out that all of them are related to mouse samples. Interestingly, there are also cancer-related datasets. Since the title of the study does not provide much information, we open the dataset summary view. Here we see that all the biological samples are subtypes of leukocyte, but nothing has been explicitly described as leukocyte, which is why we haven't found anything with the text-based search.